Welcome back to The Explainer. I'm so thrilled you're joining me today because we are setting the stage for a truly fascinating microscopic journey. We are zooming all the way in to look at the plasma membrane. Now, historically, you might have heard this referred to as the plasmalemma, the cytoplasmic membrane, or maybe just the good old cell membrane. But honestly, whatever name you prefer, this microscopically thin layer is hands down one of the most brilliant architectural marvels in all of biology. So get comfortable and let's unravel exactly how this dynamic security system manages to sustain life as we know it.
Okay, let's dive into this. Here is our roadmap for today. We're going to cover 1. What the plasma membrane actually is. 2. Its main components. 3. The unique nature of phospholipids. 4. The famous fluid mosaic model. And 5. The membrane's key functions.
All right, part 1. What exactly is the plasma membrane?
Think about it. Every single cell faces this fundamental problem. It absolutely must have a boundary. It needs something to separate its interior organelles and cytoplasm from the totally unpredictable outside environment. You can think of the plasma membrane as the ultimate security guard. It's the outermost layer of an animal cell, acting as this highly intelligent, dynamic barrier. Its primary job? To protect all the delicate interior parts of the cell from the extracellular environment and keep things perfectly organized. But it doesn't just protect, it actually gives the cell its proper shape and maintains its homeostasis. Basically, without this outer layer, a cell simply couldn't exist.
Moving right along to part 2, the main components of the membrane.
So, we know it's a security wall. But if we zoom in microscopically, what is it actually built out of? Well, fundamentally, the plasma membrane is a lipid bilayer. That means it's made of 2 layers of fats. But, as you probably know, not all lipids are the same. The vast majority, right around 50%, are phospholipids. A tiny little fraction, just roughly 2%, are glycolipids. And filling in the rest of the gaps, making up nearly 48%, are sterols, particularly cholesterol. Now that cholesterol is absolutely crucial because it embeds itself right within the layers to maintain the membrane's fluidity and elasticity, adapting on the fly as the temperature of the environment fluctuates. But hey, the wall isn't just made of fat. Embedded within this lipid layer are some highly essential proteins. On one hand, we have integral proteins. These are strongly bound and deeply embedded right into the membrane. Because they literally cut through the layer, they act as essential transporters, physically moving materials in and out. On the other hand, we have peripheral proteins. Just like the name implies, these sit right on the periphery, the outermost layer. They're only loosely attached, they don't dig deep into the membrane. Instead, they act as enzymes and communicators, facilitating all those necessary interactions between the cell and its outside environment. Which brings us to part 3, the totally unique nature of phospholipids. So, we just established that phospholipids make up half of the membrane's lipids. But how do these specific molecules actually arrange themselves to form a functioning protective barrier in a body that is mostly water? To answer that, we need to de-jargonize one of my favorite scientific terms, amphipathic. An amphipathic molecule is simply one that contains both a water-loving part, which we call hydrophilic, and a water-fearing part, which we call hydrophobic. This kind of split personality of the phospholipid is the master key to how the cell membrane can exist in a watery environment. Let's look at how that amphipathic nature physically organizes the molecule in reality. The molecule has a head made of a phosphate group. This head is negatively charged, it's hydrophilic, and it absolutely loves water. Attached to it are 2 tails made of fatty acid chains. Now these tails are hydrophobic, they literally hate water. So when you put a bunch of these molecules in an aqueous environment together, they naturally form a double layer. The water-loving heads face outward toward the extracellular environment and inward toward the watery cytoplasm. Meanwhile, the water-fearing tails hide tightly tucked on the inside, sandwiched safely away from the water. It's brilliant. This natural arrangement automatically creates a tightly packed, tightly sealed bilayer.
Let's move to part 4, the fluid mosaic model.
Now that we've got all our building blocks laid out, our phospholipids, our cholesterol, our integral and peripheral proteins, how in the world do they all fit together in a living animal cell? To explain this, scientists Singer and Nicholson proposed what is now considered the absolute gold standard biological model for the plasma membrane. They called it the fluid mosaic model. Let's break down that wonderful name, because it's actually perfectly descriptive. Why fluid? Well, because the membrane is absolutely not a rigid, solid brick wall. Thanks to components like that cholesterol we talked about and the shifting phospholipids, the membrane actually has flexibility and elasticity. It bends, it moves. And why mosaic? Because much like a mosaic piece of art made of all those different colored tiles, the membrane is a dynamic patchwork of diverse parts: lipids, proteins, cholesterol, carbohydrates, all floating and shifting together. It's a constantly moving, incredibly complex biological tapestry.
Okay, let's wrap things up with part 5, the key functions of the plasma membrane.
We've seen its microscopic architecture, so let's zoom back out for a second and look at how this incredible structure performs vital biological tasks. The membrane has a few massive responsibilities. First and foremost, obviously, is protection. It keeps harmful toxins and pathogens strictly out. Second is recognition and cell signaling, which is where those peripheral proteins and glycolipids come into play, allowing the cell to basically talk to its neighbors. But the third function is arguably its most fascinating trick: selective permeability. Because the membrane's interior is packed with those water-fearing fatty acid tails, it acts as an incredibly intelligent filter. It doesn't just let anything pass through. So, how does selective permeability actually work in practice? Well, it entirely depends on the molecule. Small non-polar molecules, like oxygen and carbon dioxide, get to slide right on through the lipid bilayer via direct diffusion, which is absolutely essential for our lungs during respiration. But larger molecules, or hydrophilic ones, like glucose, they can't just wander through that tightly packed wall. No way. They require VIP access. They have to be escorted across by those deeply embedded transport proteins we mentioned earlier. But what if a molecule is truly massive? Or what if the cell needs to swallow a huge quantity of material all at once? A transport protein is way too small for that. Well, the cell uses a really cool process called endocytosis. Let's walk through the steps here. Step 1. The flexible cell membrane actually extends outward and approaches the large material. Step 2. Because of its highly fluid nature, the membrane literally folds in on itself, wrapping around the material to form a little protective pocket called a vesicle. Step 3. This vesicle pinches off and safely transports the massive cargo right inside the cell. It's literally the membrane swallowing the material whole. And naturally, the exact reverse process happens when the cell needs to take out the trash. We call this exocytosis. Step 1. Inside the cell, a vesicle forms around the bulky waste material. Step 2. This little package moves toward the outer plasma membrane. Now, because the vesicle and the membrane are made of the exact same lipid materials, they just seamlessly fuse together. Step 3. The membrane opens up and the waste is released outside the cell into the extracellular space. The membrane acts as this completely dynamic, shifting gateway.
Which kind of leaves us with a profound realization, right? This delicate, constantly moving patchwork of fats and proteins has the robust architecture required to let a cell breathe, eat, excrete, and protect itself. It truly is the ultimate security system, but it also leaves us with a pretty provocative question. If this incredibly intricate fluid barrier is the very foundation of how a cell survives, how might unlocking its final secrets allow us to cure cellular diseases, or maybe even engineer totally new forms of microscopic life down the line? I hope this explainer has left you marveling at the plasma membrane and super eager to explore the endless wonders of cellular biology. Thanks for hanging out with me, and keep learning.